Hello, everybody. Thank you for taking the time to register for and attend this webinar today. My name is Dustin Fan. I am an oncology analyst with Datamonitor Healthcare, and today we'll be discussing PD-1, PD-L1 inhibitors, the first major wave of immuno-oncology. So this is our agenda for today. First, we'll take a brief overview of the current immuno-oncology market, and we'll take a look at some of the ongoing developments in the PD-1, PD-L1 therapy space. Now, some of the key therapies we'll be discussing today include Opdivo, Keytruda, Tecentriq, Imfinzi, and Bavencio. We'll also take a look at some of the challenges associated with PD-1, PD-L1 inhibitor development and treatment. And to provide a little bit of context, we'll also take a look at two indications that have seen a lot of the challenges, but also experienced a lot of development over the past year: non-small cell lung cancer and bladder cancer. And finally, we'll look forward to the future of immuno-oncology. Now, as we go along, please go ahead and write down any questions you might have. And at the end of the presentation, I've included a slide with my email information on it. And you can email me with any questions. This will allow me to answer your questions with the most accurate and up-to-date information possible. And it will also hopefully allow me to get to everyone's questions as well. So in the most basic sense, immuno-oncology is a field of medicine that focuses on the development of therapies that improve the body's ability to generate an immune response against cancer. And the general strategy here is to counteract the mechanisms that tumors use to suppress the immune system. And while a variety of mechanisms of action have been investigated, some of the key classes within the immuno-oncology space include cancer vaccines, oncolytic viruses, bispecific T-cell engagers, adoptive T-cell therapy, the most well-known example of which is chimeric antigen receptor T-cells, and then, of course, our immune checkpoint inhibitors, which include the CTLA-4 inhibitors, such as Yervoy or tremelimumab, and then the PD-1, PD-L1 inhibitors. And it's this last class, the PD-1, PD-L1 inhibitors, that have garnered the most interest in recent years. PD-1, PD-L1 inhibitors operate within the PD-1 pathway, which under normal conditions downregulates the immune system and suppresses T-cell activity. So again, under normal conditions, this guards against autoimmunity. It reduces immune response after disease elimination, and it prevents damage from massive inflammatory response. Now, it's been hypothesized that PD-L1 overexpression can contribute to tumor invasion, or it will allow the tumors to escape the immune system. And this is simply because the host immune system recognizes the tumor as self due to the PD-1, PD-L1 interaction. The primary difference between the PD-1 and PD-L1 inhibitors is that one blocks from the receptor side, which is the PD-1 inhibitors, and the other blocks from the ligand side, which is the PD-L1 inhibitors. PD-L1 is also expressed on tumor-infiltrating immune cells, which include, for example, macrophages or dendritic cells. But at this moment in time, there's not really a clear advantage to targeting one portion of the pathway over the other. So here we have a timeline of FDA immunotherapy approvals from between about August 2010 until about now. So in August 2010, Provenge was approved for the treatment of prostate cancer. And Provenge is interesting in that it was a custom immunotherapy. It was made with autologous dendritic cells. So the dendritic cells are taken from the patients, processed, and reinfused back into the patients with the hopes of stimulating the immune system to better target the cancer. And as you can see, there's kind of a gap period in terms of FDA immunotherapy approvals all the way until about March 2011, when Yervoy was approved for melanoma. And this made Yervoy the first immune checkpoint inhibitor approved for oncology. And more specifically, it is a CTLA-4 inhibitor. And again, as you can see, there's a little bit of a gap in terms of FDA immunotherapy approvals, all the way until about September 2014, when Keytruda was approved for second-line melanoma. And this made Keytruda the first PD-1, PD-L1 immune checkpoint inhibitor approved for oncology. And since then, there's been a continuous flurry of development and approvals. So as you can see, while we see other mechanisms such as bispecific T-cell engagers like Blincyto or Imlygic, an oncolytic virus, or most recently Kymriah, a CAR T-cell therapy, the majority of FDA immunotherapy approvals is within the PD-1, PD-L1 therapy space. Some of the key initial FDA approvals for PD-1, PD-L1 inhibitors include Keytruda for melanoma, again, in September 2014, Opdivo for melanoma in December 2014, Tecentriq for urothelial bladder cancer in May 2016, Bavencio for Merkel cell carcinoma in April 2017, and finally Imfinzi for urothelial bladder cancer in May 2017. Here we have a graph that shows some of the key mechanisms that are undergoing development within the immuno-oncology space. Now, this data is dated to the end of June 2017. However, the key trend is still relevant and it's still accurate. So out of the 1,800 or so currently ongoing immuno-oncology trials, as you can see, about 928 of them are within the PD-1, PD-L1 therapy space. So PD-1, PD-L1 therapies are the lead mechanism. Second are the CTLA-4 inhibitors, such as Yervoy and tremelimumab. Then, of course, we have the CAR T-cell therapies, as well as IDO inhibitors and oncolytic viruses. Now, it's worth noting that there's likely to be some overlap due to combination trials as well. However, as you can see, PD-1, PD-L1 inhibitors have the highest amount of investment relative to other immuno-oncology mechanisms. For a greater level of granularity, we can look at the top 10 products in terms of ongoing immuno-oncology trial count. Now, note that all 5 of the major PD-1, PD-L1 inhibitors are included in this list. Again, these include Keytruda, Opdivo, Imfinzi, Tecentriq, and Bavencio. We also have the 2 CTLA-4 inhibitors, Yervoy and tremelimumab, which are also being developed for use in combination with PD-1, PD-L1 inhibitors. We also have PDR001, a PD-1 inhibitor from Novartis, as well as epacadostat, an IDO inhibitor, and CTL019, again, a CAR T-cell therapy. Rather expectedly, Keytruda and Opdivo lead the race with 317 trials and 281 trials, respectively. Though, in all likelihood, this number is probably larger by now. And this is due to their early entry into the market, as well as their subsequent success and their continued ongoing development. In terms of the specific indications for development is taking place, here we've listed the top 10 cancers in terms of ongoing PD-1, PD-L1 clinical trials. You can see that all 10 of these indications are solid tumor indications. This is where PD-1, PD-L1 therapies were first approved. This is where they saw regulatory and commercial success, and this is where they continue to be developed. And this is largely influenced by the fact that solid tumor indications as a whole have a higher incidence and prevalence than rarer hematological malignancies. And so there's potential for significant commercial reward. As you can see, the top 3 cancers in terms of ongoing PD-1, PD-L1 clinical trials include non-small cell lung cancer, melanoma, and breast cancer. If we took a look at the development status of key PD-1, PD-L1 agents in oncology, we again see that a significant proportion of the indications for which these drugs are approved or ongoing development are for solid tumor indications. Though, as you can see, there's clearly still development in hematological malignancies as well. Now, please note that this list is dynamic, right? There's new trials starting all the time in new indications as well as new agents. And the data here is dated, though, to just last week, September 7th, 2017, so it's recent. As you can see, Opdivo and Keytruda lead with approvals in 7 distinct tumor indications each. These 2 drugs also have BLAs under review for a number of new indications as well. Tecentriq and Bavencio are each approved in 2 indications, and Imfinzi is approved in 1. But they do have phase 3 trials in other indications, suggesting new approvals could be coming. And a large number of ongoing trials in all these different cancer types suggest that there are still opportunities for the PD-L1 inhibitors, Tecentriq, Imfinzi, and Bavenci, which came after the PD-1 inhibitors, to establish themselves, often in areas with limited competition and high unmet need. And while PD-1, PD-L1 therapies were initially approved as monotherapies, there's now a trend towards studying their safety and efficacy in combination trials. Opdivo and Keytruda currently lead in terms of the number of combination trials, again, owing to their early entry into the market and their subsequent success. The largest proportion of Keytruda's combination trials, the very last row, are with chemotherapy, shown here in pink. But a large proportion of trials are with targeted therapies as well, shown there in dark purple. And some of these targeted therapies include tyrosine kinase inhibitors, such as Giletrif, or monoclonal antibodies, such as Cyramza or Herceptin. In contrast, Opdivo has a large proportion of combination trials with other immuno-oncology products, the majority of which are likely to be with Bristol Myers Squibb's own CTLA-4 inhibitor, Yervoy. And this combination first saw success in melanoma. Similarly, Imfinzi has a large number of combination trials with other immuno-oncology products, likely AstraZeneca's own CTLA-4 inhibitor, tremelimumab. So this particular combination, Imfinzi-tremelimumab, is expected to compete directly with Opdivo plus Yervoy in a number of indications, as well as treatment settings. Thus far, 2 immunotherapy combinations have found regulatory success in the United States. Opdivo plus Yervoy gained an accelerated approval in 2015 for BRAF V600 wild-type unresectable or metastatic melanoma, and this was later expanded in 2016 to include BRAF V600 mutation-positive patients as well. On the other hand, Keytruda plus chemotherapy gained an accelerated approval in 2017 for first-line metastatic non-squamous non-small cell lung cancer. For Opdivo plus Yervoy, Bristol Myers Squibb and Ono Pharmaceutical released data from the Checkmate 067 trial in April 2017. And this will, in all likelihood, pave the way for a full approval in first-line advanced melanoma. Now, the results from this trial revealed that the Opdivo plus Yervoy combination conferred a superior overall survival advantage compared with Yervoy alone. The Opdivo alone data was not yet available. However, there was also an increase in toxicity in the Opdivo plus Yervoy arm compared with the Opdivo alone arm. So the frequency of grade 3 and 4 adverse events was 58% in Opdivo plus Yervoy arm, compared with 21% in the Opdivo arm. And so despite the survival advantage provided by Opdivo plus Yervoy, as you can see, there's still a potential for increase in associated toxicities. Now, the data that led to Keytruda's accelerated approval in the first-line setting comes from the Keynote-021 trial, specifically cohort G. And the data revealed that progression-free survival was improved in the Keytruda arm compared with the chemotherapy-only arm. And at that time, the overall survival was not significant, simply because the median overall survival was not reached in either group. However, the Keynote-021 cohort G data was most recently updated just this past weekend at ESMO, and it revealed that improvements in overall survival were indeed significant, though the median overall survival was not yet reached in the Keytruda plus chemotherapy arm. As with the Opdivo plus Yervoy combination, there's also an increase in toxicity in the Keytruda plus chemotherapy arm compared with chemotherapy alone. But the full phase 3 data for the Keytruda plus chemotherapy combination in first-line setting is expected sometime this year. It'll be from the Keynote-189 trial, which is a phase 3 trial. Now, at this point, we can't really compare these 2 immunotherapy combination approaches. As you can see here, we have 2 different indications, 2 different levels of study. One is a phase 3, one is a phase 1/2. But it does give us insight into the potential benefits of combination therapy, as well as some of the drawbacks. Together, these data suggest that Monotherapies will continue to be an important treatment option for patients who can't tolerate additional toxicities. And uptake will probably be based also on preferred dosing schedule, cost, as well as physician comfort and familiarity. But this still does represent an important advancement in immunotherapy development. Now, despite all the advancements that have been made in the PD-1, PD-L1 therapy space, there are still challenges pertaining to PD-1, PD-L1 therapy development and treatment. And one of these challenges is drug differentiation. Physicians want to choose the best drug for their patients, but if multiple drugs are allowed or approved, how does one differentiate, right? If one drug has better safety or efficacy for a particular patient group, then ideally that is the one that is selected for the patient. But if not, it often boils down simply to access. Similarly, developers that move into competitive markets need to find a way to differentiate their products. So there's a few ways for these drugs to be differentiated. First, they could try to achieve a first-to-market advantage. And this allows the drug to enter an indication with limited competition. This means that differentiation really isn't yet needed. And this could result in greater physician comfort and familiarity over the long term. Another approach is to target new indications. They can look for opportunities in niche markets of smaller indications. So an example of this is Bavencio, which was approved for Merkel cell carcinoma. Now, Bavencio was one of the last of the PD-1, PD-L1 inhibitors to gain regulatory approval. And still yet, by entering the Merkel cell carcinoma market, it has now the first-to-market advantage, which helps ensure uptake. Another approach for differentiation is through combination therapy. Monotherapies may be similar in safety and efficacy, so sometimes there's very little differentiation. But these combinations have the potential for improved efficacy and commercial potential. However, it does also introduce new variables. So combinations can add safety and toxicity concerns. And there may also be cost concerns as well. And some of the examples of combination being investigated right now include Imfinzi plus tremelimumab, Tecentriq plus chemotherapy, Tecentriq plus Avastin, Keytruda plus Cyramza. There's many others under development. Another approach is to target unique patient settings by offering new treatment options in areas of little competition or high unmet need. So an example of that is actually Bavencio, which is currently under phase 3 development for stage 4 urothelial carcinoma in the maintenance setting. Now, this is interesting because the NCCN treatment guidelines don't actually provide much specific guidance on the maintenance treatment of metastatic bladder cancer, right? And so if Bavencio, if its phase 3 data in this treatment setting is positive, if it shows a benefit with acceptable toxicity, again, this is an area of little competition, which ensures the drug's uptake. And finally, last is to use biomarker strategies. Probably the most well-known example here is Keytruda, which was initially approved for PD-L1 positive second-line non-small cell lung cancer, which differentiated it from, say, Opdivo. It's also recently received an accelerated approval for microsatellite instability high or DNA mismatch repair deficient tumors, and this made it the first targeted therapy to get an approval based on a biomarker rather than a specific tumor location. Opdivo was also recently approved for microsatellite instability high or DNA mismatch repair deficient tumors, but this was limited only to metastatic colorectal cancer. An additional consideration for developers is the proper selection of clinical trial endpoints. Now, developers want to select endpoints that not only help bring their drug to market in a timely or expeditious manner, but it's important that physicians have the data to make the best decisions possible for their patients. And so here I highlight 3 commonly used clinical trial endpoints in oncology: overall response rate, progression-free survival, and overall survival. So for overall response rate is advantageous in that it can be measured in single-arm trials. And the metric is unique to that drug. Not only that, the time scales necessary to measure overall response rate are really quite short. However, it's not really a comprehensive measure of drug activity. It doesn't always translate well into progression-free survival or overall survival. Now, progression-free survival is advantageous in that it measures stable disease. It's sometimes a surrogate consideration for quality of life, and again, it typically requires shorter timescales. However, the measure of progression is somewhat imprecise, and this can be exacerbated by pseudoprogression, which is the tumor or radiographic progression without clinical deterioration. Not only that, progression-free survival requires frequent assessment, and it's really quite a weak surrogate for survival. And then, of course, we have overall survival, which is something of the gold standard, right? It is a direct measure of, or it is a measure of direct benefit. However, the timescales necessary to measure overall survival are really quite long, and this can delay bringing a drug to market. It also considers unrelated deaths, and it can be affected by subsequent therapies. So on the right, you can see the primary endpoints for phase 2 and 3 trials in terms of trial count. This is specific to PD-1, PD-L1 therapies. For the phase 2 trials, you can see that the majority of trials use overall response rate as a primary endpoint for the reasons that we just discussed, right? It can be measured in single-arm trials, and they can be assessed on shorter timescales. And indeed, a lot of PD-1, PD-L1 inhibitors have gained accelerated approvals based on overall response rate. However, when better measures of drug activity, more comprehensive measures of drug activity are required, then overall survival or progression-free survival are going to be more commonly used, as in phase 3 trials. Another challenge for drug developers is the use of PD-L1 as a biomarker. Now, it was initially hypothesized that PD-L1 expression might be an indicator of response to PD-1, PD-L1 inhibitors. And this is because, as we briefly discussed earlier, PD-1, PD-L1 interaction downregulates the immune response. Right? And so a correlation between PD-L1 expression and response has been observed, but the results have been somewhat inconsistent. We've seen benefits for both PD-L1 positive and PD-L1 negative tumors, and in some cases, complete responses can't be excluded for PD-L1 low or even PD-L1 negative tumors. In addition, PD-L1 expression is dynamic. It's expressed on multiple cell types to include, for example, tumor cells, immune cells, or elsewhere in the tumor microenvironment. It can also be expressed differently based on disease site, and expression can vary depending on the time of measurement. So studies have suggested that prior chemotherapy can cause tumors to become inflamed, which may increase PD-L1 expression, for example. In addition, PD-L1 expression is not binary. It occurs on a sliding scale. So in some cases, PD-L1 expression greater than or equal to 50% is considered PD-L1 positive. In other cases, it might be greater than or equal to 10%, or 5%, or 1%. However, patients can even test differently depending on the assay that is used. So to better illustrate the challenges of PD-L1 as a biomarker, here we have the 5 key PD-1, PD-L1 inhibitors in development for or approved for NSCLC, non-small cell lung cancer. On the far left column, we have the 5 drugs: Keytruda, Opdivo, Tecentriq, Imfinzi, and Bavencio. And just to the right of that, we can also see the developers for each of the accompanying diagnostics, as well as the platform that is used. As you can see, the assay antibody clone for each of the diagnostics is different, as is the basis for scoring. So Keytruda, Opdivo, Imfinzi, and Bavencio all use tumor cell staining, whereas Tecentriq uses tumor cell or tumor infiltrating immune cell staining as well. And as you can see, the cutoff values can also differ between the products. And this is important because it determines whether a patient is eligible for treatment with a certain product or not. But even for the same cutoff value, there's a difference in the patients who test positive for PD-L1 expression. Let's consider, for example, a cutoff value is greater than or equal to 1%. Okay. At the very top, we have Keytruda at a cutoff value of greater than or equal to 1%. 66% of patients qualify for treatment. However, for that same cutoff value, greater than or equal to 1%, we can see that Bavencio, the very, very bottom row, 79% of patients qualify for treatment. And this PD-L1 expression testing is complicated by the fact that patients can test positive for expression with one test, but not another. And here we have the results of the Blueprint PD-L1 IHC assay comparison project. The study took samples from 38 patients and tested them with 4 of the assays from the previous slide. On the left, you can see that in terms of tumor cell staining, the assays for Keytruda, shown in black, Opdivo, shown in red, and Imfinzi, shown in yellow, were all closely aligned. There's a significant amount of overlap. However, Tecentriq in light blue was consistently lower. Now on the right, we can see the consensus areas for the 4 different assays. Okay. Tecentriq's assay, shown in the dark blue oval, classified 30 out of 38 patients as PD-L1 positive. Keytruda's, shown in dark purple, had 26 out of 38 being PD-L1 positive. Opdivo's, shown in orange yellow, had 26 out of 38 being PD-L1 positive. And Imfinzi's in green showed 20 out of 38 being PD-L1 positive. Now, interestingly, Tecentriq's assay, again in dark blue, confirmed the highest number of PD-L1 positive patients, 30 out of 38. But on the left, as we saw, Tecentriq consistently scored lower in terms of tumor cell staining. And this is probably because the assay also accounts for tumor-infiltrating immune cells. The takeaway, though, is that 19 out of 38 patients were classified as PD-L1 positive by all 4 tests, and 5 out of 38 were classified as PD-L1 negative by all 4 tests. And the remaining 14 patients had discordant results, meaning they would have had a different classification depending on which assay that was used. And this is important because if PD-L1 expression is going to be used to select patients that will best respond to therapy, then consistent and accurate testing would be beneficial. And until a common standard is established, measuring PD-L1 expression reliably is going to be a challenge. So one indication that really highlights many of the advancements as well as challenges associated with PD-1, PD-L1 therapies is non-small cell lung cancer. As you can see, again, if we revisit the top 10 cancers in terms of ongoing PD-1, PD-L1 clinical trials, non-small cell lung cancer is the leading indication in terms of ongoing trials, meaning this is the area where the most development is currently happening. As you may know, worldwide, lung cancer is the leading cause of cancer death, and non-small cell lung cancer accounts for about 85% of lung cancer. So in 2016, there were an estimated 522,730 incident cases of non-small cell lung cancer in the US, Japan, and 5 major EU markets. And for the purpose of our analysis, these 5 major EU markets are France, Germany, Italy, Spain, and the UK. And the number of incident cases is predicted to increase to 684,750 cases by 2035. Another way to look at this is to take a cross-sectional view of the NSCLC patient population. Patients who are actively treated can be divided into various stages of disease. Now, these include stage 1 and stage 2, which are your early stage patients, stage 3, which are your locally advanced patients, stage 4, your metastatic, and of course your local and distant relapse patients. These patients can be further divided into non-squamous and squamous histologies. And then the non-squamous patients can be divided into EGFR mutation positive, ALK rearrangement positive, or patients without either of these addressable mutations, which we've simply called here other non-squamous. And finally, each of these populations can be divided into individual lines of therapy. Now note that a significant proportion of NSCLC patients have advanced disease, specifically stage 3 or greater. And for these patients, curative surgery is just no longer an option. So treatment in this setting relies primarily on pharmaceutical interventions. If we take a look at the prognosis of non-small cell lung cancer, we can see that NSCLC has among the worst 5-year survival rates of all cancers, with only 18.1% of patients surviving 5 years or more after their initial diagnosis. And this is predominantly due to the fact that 57% of patients are diagnosed at the distant stage, at the metastatic stage, which has a mere 5-year survival rate of 4.5%. So it's this increasing prevalence of NSCLC combined with a high proportion of patients at late-stage disease where patient outcomes are really quite poor that make this an area of unmet need and really contributes to the interest in development for new therapies in this setting. PD-1 inhibitors were first introduced to the NSCLC market in 2015. Opdivo first gained regulatory approval in the US, EU, and Japan in 2015, where it was approved for second-line metastatic non-small cell lung cancer. Keytruda was initially approved in the US in 2015, and then in the EU and Japan in 2016. And it was approved for PD-L1 positive second-line metastatic NSCLC. Now, Merck and Co. defined PD-L1 positive initially as PD-L1 expression greater than or equal to 50%. This really highlights the respective developer strategies. With Opdivo, Bristol Myers Squibb and Ono wanted to target the widest possible patient population that would benefit from therapy. And this ensures maximal uptake. With Keytruda, however, Merck and Co. sought to identify the patient population that they believed would best respond. But this has the effect of potentially severely limiting uptake. Soon after Keytruda and Opdivo were approved for previously treated patients, they were in a race for the first-line setting. In June 2016, Merck's KEYNOTE-024 study showed a progression-free survival and overall survival benefit for Keytruda in first-line NSCLC compared with chemotherapy. And this was limited to PD-L1 positive patients. Again, patients with PD-L1 expression greater than or equal to 50%. But in August 2016, Opdivo failed its first-line CheckMate 026 trial. And then beginning in October 2016, Keytruda gained regulatory approval for the first-line setting in the US, Japan, and EU pretty much in quick succession. In Japan, it received first and second-line approval simultaneously. And then in May 2017, Keytruda plus chemotherapy received an accelerated approval in the US for the first-line setting. And this was not limited by PD-L1 expression. All is not lost for Opdivo. However, Opdivo plus Yervoy showed promising results in the first-line setting in the phase 1b CheckMate 012 trial. And they have a phase 3 CheckMate 227 trial in the first-line setting that is expected to read out in 2018. The PD-L1 inhibitors were approved for NSCLC shortly after Keytruda and Opdivo. The PD-1 inhibitors gained regulatory approval. Tecentriq was the first of the PD-L1 inhibitors to gain approval, and it was successfully given approval in 2016 in the U.S. for second-line metastatic NSCLC. It recently received a positive opinion from the EMA, so EU approval is expected rather soon. And Chugai filed for Japanese regulatory approval in Q1 of 2017. Imfinzi actually has 3 phase 3 trials that are reading out this year in 2017. Now, Imfinzi showed promising results in the Pacific trial, which studied its use following chemoradiation in stage 3 patients. In the Mystic study, however, the drug failed to show a progression-free survival advantage over chemotherapy. Now, AstraZeneca may still try to gain regulatory approval in the first-line setting based on overall survival data, but this isn't due until the first half of 2018. And the last Imfinzi trial in NSCLC is expected to read out this year is the Arctic trial, which is testing the drug in the third-line setting. And approval here would allow the drug to minimize direct competition with Tecentriq, Keytruda, and Opdivo and gain some uptake that way. And finally, Bavencio is expected to be the fifth PD-1, PD-L1 inhibitor to gain regulatory approval, and its phase 3 data is expected to read out sometime in 2018. This is the current competitive landscape for non-small cell lung cancer in the U.S. It shows current approvals in dark blue, label expansions in green, pipeline therapies in yellow, and areas of significant off-label use in light blue. This area in the red box is where the majority of PD-1, PD-L1 inhibitor uptake will occur. Now, this includes squamous patients, as well as non-squamous patients without addressable mutations. Now, again, these addressable mutations include ALK rearrangement positive patients, as well as EGFR mutation positive patients. Now, the ALK positive and EGFR positive patients are treated primarily with small molecule inhibitors, tyrosine kinase inhibitors, but they still represent a minority of the total NSCLC patient population. That doesn't mean that PD-1, PD-L1 inhibitors won't be able to get uptake in the EGFR positive and ALK positive patients. Prescribing labels for the PD-1, PD-L1 therapies require that patients first be treated with targeted therapies. But this essentially allows for patients who don't respond to targeted agents to then be treated with PD-1, PD-L1 inhibitors. Owing to their broad applicability, PD-1, PD-L1 targeted therapies are going to be the key driver for NSCLC market growth. We project that sales of key therapies in NSCLC will grow from $7.8 billion in 2016 to over $20.8 billion excuse me, by 2025 in the US, Japan, and 5 major EU markets, with PD-1, PD-L1 inhibitors accounting for over half of total sales. Of the PD-1, PD-L1 therapies, we project that Keytruda is going to be the leading drug in its class by 2025, owing to its first-line approvals. Now, Opdivo had strong sales in 2016, but the drug's failure in the Checkmate 026 trial means that it missed the lucrative first-line setting. And so approval for the first-line setting is going to be delayed until probably 2018. Also, there's increasing competition in the second-line setting now with Keytruda and Tecentriq. As a result, Opdivo sales are going to be impacted by this failure going into 2017, but with first-line approval in 2018, we think the drug is going to recover and be among the highest selling. Now, Tecentriq was originally projected to have stronger performance. It was on track to be the first PD-1, PD-L1 therapy to be approved with chemotherapy. But Keytruda's accelerated approval is going to have a negative impact on that. Still, we expect fairly strong sales. Bavencio and Imfinzi, though late to the NSCLC market, are still expected to have some uptake since they are being developed for fairly unique settings. As we saw earlier, Imfinzi, besides targeting the first-line setting, will also target stage 3 patients who have undergone concurrent chemoradiation, as well as the third-line setting, whereas Bavencio is going to use a biomarker strategy, and it's going to target PD-L1 positive patients. Besides non-small cell lung cancer, I also want to talk a little bit about bladder cancer as well. This is an exciting time for bladder cancer. Prior to May 2016, there were no new therapies approved for bladder cancer in nearly 30 years. But since then, basically in just the last year, the drug has seen a flurry of development in terms of approvals, as well as some setbacks as well. Again, if we take a look at the top 10 cancers in terms of ongoing PD-1, PD-L1 clinical trials, you can see that bladder cancer is among these top 10. Like non-small cell lung cancer, bladder cancer is attractive due to its growing incidence. Bladder cancer is the 5th most common cancer in the US and Europe, and the 9th most common cancer worldwide. And about 90% or so of bladder cancers are urothelial carcinomas. And so urothelial bladder cancer is going to be the emphasis of our talk today. In 2016, there were an estimated 230,980 incident cases of bladder cancer in the US, Japan, and 5 major EU markets. Again, for the purpose of our analysis, these 5 major EU markets are France, Germany, Italy, Spain, and the UK. And the number of incident cases is predicted to increase to over 327,060 cases by 2036. Again, we can take a cross-sectional view of the bladder cancer patient population. The actively treated patient population can be broken down into various stages of disease to include non-muscle invasive, which are your stage 0 and 1 patients, your muscle invasive, which are your stage 2 and 3 patients, your stage 4 patients, which are your metastatic patients, and then of course your local and distant relapse again. Now, a large proportion of these patients, as you can see, are in early stage. So for such patients, surgery is the most commonly used modality of treatment. There are a lower percentage of patients, compared to NSCLC at least, with metastatic disease. These patients don't typically benefit much from surgery and are thus treated with pharmaceutical interventions. So as a result, increasing prevalence of urothelial bladder cancer, combined with the lack of new therapies for patients with advanced disease, make this an area of high unmet need. So when Tecentriq gained an accelerated approval for the treatment of second-line urothelial bladder cancer in May of 2016, this made it the first new therapy approved for bladder cancer in nearly 30 years. And it remained uncontested all the way until starting in February 2017. Opdivo, Imfinzi, Bavencio, and Keytruda were all approved for the second-line setting in rapid succession. Basically, in a one-year period, all 5 PD-1, PD-L1 inhibitors were approved for the same patient setting in bladder cancer, though Keytruda was the only one with a full approval. In addition, Tecentriq and Keytruda have accelerated approvals for first-line cisplatin-ineligible patients. Since then, Opdivo and Keytruda have also gained European regulatory approval, but in May 2017, Tecentriq failed the phase 3 IMvigor 211 study. Now, the phase 3 IMvigor 211 study was meant to be a confirmatory study that would convert Tecentriq's accelerated approval in the second-line setting into a full approval. However, the drug failed to show an overall survival advantage compared with chemotherapy in second-line platinum-treated patients. And while the full data isn't out yet, it appears that Roche and Chugai faced some of the challenges common to the PD-1, PD-L1 therapy class. For one, the drug received an accelerated approval based on overall response rate. But as we discussed earlier, overall response rate doesn't always translate well into other measures of efficacy, such as overall survival. Not only that, the results in the Tecentriq-treated patients in the IMvigor 211 study were said to be consistent with the phase 2 IMvigor 210 study, which makes the failure all the more surprising. Again, we don't know what the cause for the failure is yet since we don't have the full data, but we might be able to speculate on the failure a little bit. Some might believe that high PD-L1 expression could help improve responses. And here we have the pivotal data for each of the 5 PD-1, PD-L1 therapies approved for urothelial bladder cancer in the second-line setting. Now, as you can see, where the data is available, complete responses could not be excluded for PD-L1 low or PD-L1 negative patients. And in many cases, the complete response rate was similar for PD-L1 high and PD-L1 low patients. And this suggests that PD-L1 expression in itself may not be the reason for failure. If we take a look at the overall patient population, we can see that the overall response rates were remarkably similar across all 5 therapies. As we previously discussed, though, overall response rate is not always a comprehensive measure of efficacy. It doesn't always translate well into other measures such as progression-free survival or overall survival. But these results did translate for Keytruda, allowing the drug to get a full approval. But they didn't translate for Tecentriq. And this is, again, surprising because the overall response rates were fairly similar. Not only that, the fact that the results of the Tecentriq-treated patients in IMvigor 211 were said to be similar to IMvigor 210 makes it even more confounding. Ultimately, we'll have to wait for further data to better understand the reasons for failure. It's worth noting, however, that the EMA adopted a positive opinion for Tecentriq in the second-line setting just recently, and it cited the IMvigor 211 study in influencing its positive opinion. So if the drug does ultimately get approved by the European Commission, it'll be interesting to see what they say, if anything, about the drug's failure in the IMvigor 211 study. Since Keytruda is the only PD-1, PD-L1 therapy to demonstrate a statistically significant survival advantage over an active comparator, it has a full approval in the US again. And combined with its first-line accelerated approval, we think this will help it become the leading drug in the PD-1, PD-L1 therapy class for urothelial bladder cancer. Now, Tecentriq was the first drug to market, but the drug's failure in the IMvigor 211 study puts that second-line approval at risk, and at the very least, it reduces confidence in the drug. It could also negatively impact the drug's ability to gain approval in other markets, but at the moment, it's also being developed for the first-line and adjuvant settings. We also think Opdivo is going to be among the highest-selling drugs. It really benefits from a lot of physician comfort and familiarity, a lot of data supporting its use from other oncology indications. And it's currently under development for the first-line setting in combination with Yervoy, as well as the adjuvant setting. Imfinzi is also expected to compete in the first-line setting, where it will seek approval with tremelimumab. And finally, Bavencio is one of the last to market. But again, that one targets the maintenance setting, which again will be interesting because NCCN treatment guidelines really don't provide much guidance on the maintenance treatment of patients with metastatic disease. As a class, PD-1, PD-L1 therapies are going to be the key driver for urothelial bladder cancer market growth in the 7 major markets. We project that sales within this market will grow from 112 million to over 4.7 billion by 2025, with about 90% of that coming from PD-1, PD-L1 therapies. And again, this is because for nearly 3 decades, there were no new therapies approved for urothelial bladder cancer. And now we have a flurry of development within the PD-1, PD-L1 therapy space. With the PD-1, PD-L1 inhibitors currently dominating the immuno-oncology space, I want to briefly take a look at the future of immuno-oncology as well and consider the next possible wave of advancements. Keytruda recently received an accelerated approval for microsatellite instability high, or DNA mismatch repair deficient tumors that progressed following prior treatment. And this makes it the first therapy approved for oncology based on a biomarker rather than a tumor location. This is a tumor agnostic approval. As we discussed, Opdivo was also approved for NSIH or DMMR. However, this was limited to metastatic colorectal cancer. Nonetheless, I think this really serves as a sort of proof of concept and could serve as the basis for further future study. Some of the other key mechanisms that have been getting a lot of attention within the immuno-oncology space include also IDO inhibitors and CAR T-cell therapies. IDO inhibitors are currently under development primarily in solid tumor indications. The data hasn't been particularly impressive in single-agent trials, but there is an interest in their use in combination with immunotherapies. And trials involving IDO inhibitors plus Keytruda and Opdivo thus far have shown some quite promising results. And then, of course, we have the CAR T cell therapies, chimeric antigen receptor T cells. And these are one of the most promising agents in clinical development for hematological malignancies as well as solid tumors. The key players here are Juno, Kite, and Novartis. And it's these CAR T cell therapies that I think are going to be the next major wave of immuno-oncology. CAR Ts are a form of customized immunotherapy made from the patient's own T cells. So the T cells are genetically engineered to express chimeric antigen receptors that target tumor-specific antigens. And this is done through the delivery of encoding genes by way of viral vectors. So essentially, the T cells are taken from the patients, they are processed, they are expanded and then reinfused back into the patient with the hopes of stimulating the immune system to better target the cancer. Now, if we take a look at the top 10 cancers in terms of ongoing CAR T trials, we see that the majority of these trials are taking place in hematological malignancies, as opposed to what we saw before with the solid tumor indications with PD-1, PD-L1 inhibitors. And what this illustrates is an opportunity for these CAR Ts to fulfill an unmet need. Now, a large proportion of these trials, as you can see, are early phase single-arm trials. And the data thus far has been pretty promising. We've seen some strong overall response rates, and in many cases, they've been fairly durable. And given that the development of these therapies are primarily in hard-to-treat hematological malignancies, some of these CAR T cell therapies are likely to receive accelerated approvals based on overall response rate, as we just saw recently with Kymriah. Here we have the development status of key CAR T cell therapies in oncology. Kymriah currently leads this class with its recent approval in relapsed refractory pediatric ALL, followed by KTE-C19 with a BLA in relapsed refractory diffuse large B-cell lymphoma. We think that JCAR017 will likely be the third of these autologous CAR Ts to reach the market. Now, this list is not exhaustive, of course. If you follow the CAR T space, you know that there are a large number of CAR T therapies in development, in phase 1 or even preclinical development. So I've only listed some of the key therapies here from the key developers. Like the PD-1, PD-L1 inhibitors, there are also some potential challenges associated with CAR T treatment and development. One of the most prominent of these concerns is safety and toxicity. The side effects of rapid early CAR T expansion include cytokine release syndrome, which is a sort of massive inflammatory response, high-grade neurotoxicities, as well as cerebral edemas. And while safety and toxicity was a major concern for all the CAR T products, one of the most well-known examples of this is JCAR015 in relapsed refractory ALL. In July 2016, the phase 2 rocket study was halted after 3 patient deaths were reported. And it was initially suggested that the fludarabine-cytarabine preconditioning regimen was the cause of this. But the hold was lifted 3 days later, and the fludarabine was pulled from the preconditioning regimen. However, in November 2016, the trial was halted after an additional 2 patient deaths, and by March 2017, JCAR015 was discontinued, and the phase 2 rocket study was suspended. Now, an additional challenge associated with CAR T development and treatment is manufacturing and logistics. Many of the CAR Ts that are currently under development or further in development are autologous CAR Ts, basically the ones made from the patient's own cells. This has a long manufacturing time, up to 3 weeks vein to vein, meaning 3 weeks from when the T cells are taken from the patient, processed and reinfused back into the patients. This is further exacerbated by the limited number of production facilities. And as a result of this, the long manufacturing time, the limited production facilities, as well as the customized nature of these CAR Ts, we're probably going to experience the high cost of production, which is going to be passed on to the payers. Despite these challenges, the outlook for these drugs is positive, and the efficacy data thus far suggests that they could play an important therapeutic role in hard-to-treat hematological malignancies and possibly solid tumor indications. So in summary, today we've discussed some of the challenges facing both physicians and developers in the PD-1, PD-L1 therapy space. These challenges include drug differentiation, careful selection of clinical trial endpoints, the use of PD-L1 as a biomarker for response, as well as the accurate and consistent measurement of PD-L1 expression. Nonetheless, PD-1, PD-L1 inhibitors have grown to become the leading class of immunotherapies in oncology since they first gained regulatory approval. They're now approved in 8 distinct tumor indications, and they're the leading class of immunotherapies in terms of ongoing development as well. Sales of PD-1, PD-L1 inhibitors have grown from $84 million in 2014 when they were first approved to over $6.2 billion in 2016. Now, looking forward to the future of PD-1, PD-L1 therapy, we can expect new combination therapies, new indications, as well as new treatment settings, and possibly further development in tumor-agnostic indications as well. And finally, we briefly discussed CAR Ts, which are among the most promising new immunotherapies under development for oncology, and I think could usher in the next major wave of immuno-oncology. Thank you very much for your time today. If you have any questions, please feel free to email me directly. Here's my email address, and I will get back to you as soon as possible. Thanks again very much for your time today.